Yeah, welcome to the Markov Modeling and PyEMMA tutorial. My name is Frank Noé. First of all, I'd like to thank very much all the people in my group that are here and that have helped to set us up. So there's Martin, Tim, Christoph, Moritz and Fabian, and there's also Luca and Andreas. I guess that was too fast to remember, but so at least you have seen them now once, and oh, and that's the end. So you can learn the names during these couple of days. Another thing I wanted to announce, so there's not, as you probably know, there's 3 days of core workshop, meaning 3 days of lectures and practical sessions, and on Thursday we will have talks. And so, just remind me, Thursday we have, it's only talks, or it's the...
Thursday morning we have talks for about 3 hours, and in the afternoon we have project phase. Project phase,
okay. So and Thursday morning we still have some room for additional talks, like up to 3 or 15 minutes. So if you're doing a project, even if it's in progress, which is somehow related to the workshop, and you'd like to talk about it, let us know. It's nothing super refined, it's work in progress, that's totally fine. Yes?
Especially if you have brought your own data and you want to introduce this kind of data, because if we know what you want to work on, then it's easier for us to help you in the project phase.
Right. Okay, so I will give you a 1 hour overview of Markov modeling. So this is mostly overview and a little bit of theoretical background. Not so much PyEMMA yet. So I will skip over this page. I think you have seen this already. Most of you have a working installation. Some of you maybe still have some troubles to get it running, especially if you're using Windows, but I hope we can manage that. Yeah, let us go into the talk. So first of all, one of the subjects we're interested in is how biological processes, how cells, for example, work. This is a cell, and if you zoom into a cell and look at a roughly 100 by 100 nanometer cube of the cytosol, then you would theoretically, if you could resolve it, be able to see something like this. And what this shows is proteins in the cytosol, so in the cellular liquid diffusing around, and you see some of them are diffusing fast, others are diffusing slow, sometimes they form contacts, and these contacts are often transient and then they dissociate again. There are some complexes that are long-lived, and so there's transport going on, most of the biochemical processes that your body is doing, so metabolism, digestion, development of pores, when you move a muscle, all of that essentially relies on biomolecules such as proteins swimming around, forming contacts, breaking contacts, and occasionally undergoing conformational changes. Conformational changes means there are structures. A protein has a certain structure, a 3D shape, but it can have multiple such structures and transition between them. And these transitions typically are rare events. So that means for a very long time, the molecule stays in a certain shape, fluctuates around this shape, and eventually flips into another shape. And these shapes are often associated with biological functions. Certain shapes can bind other binding partners, other proteins, for example. Others can't. And so this diffusing around, the switching of the shapes, and the association and dissociation, those are, in some sense, the basic elements that implement biological function on a cellular level. Now, all of these levels, so these microscopic dynamics and the macroscopic dynamics of the cell and the even more macroscopic dynamics of the body are coupled with one another. In other words, if you make little changes here, you can affect the entire cascade of scales. If you make a mutation in a protein, you can distort the equilibrium between the different shapes the protein can switch between, and as a result, you will change certain biochemical pathways, meaning some things will not happen anymore or will happen too strongly, and as a result you can get a disease, for example, or the global behavior can change. And because of this coupling from the microscopic to the macroscopic scales, it is sometimes at least important to understand the smallest scale, to understand how protein structures or other biomolecular structures look like, so what these 3D structures are, and how they switch between each other, and what the equilibrium between these different structures is, so what is the probability of being here or here, and how would it change if I make a little change to the system, like a mutation to the protein. Okay, and this is why we do molecular dynamics. The problem here is, well, first of all, the problem is experimentally, this is very hard to observe. All of this, with this like 100 nanometers, is below the diffraction limit of optical light. So in other words, we cannot really build a microscope to resolve this. So this is smaller than 1 pixel in a light microscope, no matter how good the optics are. We can use higher frequency light, such as X-rays, but higher frequencies have more energy, so that's very destructive to biological matter. And for this reason, we need to do a lot of extra work to get images before the material is destroyed. For example, taking a lot of copies of the same protein and putting them in a crystal and keeping it very cold, and then we can get a diffraction limit, a diffraction pattern with X-rays from which we can infer a structure. But all of this is very indirect and it's not happening at the conditions at which cells like to operate. So that's one of the reasons why we use simulations. Unfortunately, simulations, if we want to resolve atomistic dynamics, are very expensive. So this is a simulation box which shows mostly water and 2 small proteins, Barnase and bar star. Barnase is a bacterial protein, and bar star is another protein which the same bacterium expresses, which is a natural inhibitor. So it can bind to Barnase and inhibit its activity, which is basically to cleave RNA. Now, this here is a simulation box which shows the 2 proteins and about 30,000 water molecules. So the entire simulation system has about 100,000 atoms. And this is pretty much the minimal size you need to simulate the association and dissociation of these 2 small proteins, because you want, when you have them separated, you still want to have like at least 1 nanometer of water around the system. Otherwise, there is a strong interaction between the periodic replicas, and that would be artificial. Okay, unfortunately, we can only simulate femtosecond time steps. And therefore, we can go to simulation lengths of microseconds and easily hundreds of nanoseconds, maybe a couple of microseconds. And so this shows you 100 nanosecond simulation, starting from the dissociated state. So the proteins are diffusing around, they start to form in contact, but they don't really bind yet. And once they bind, then they really stay there very long. This particular complex has a lifetime of about 1 hour in equilibrium. So it's basically really hopeless to simulate the dissociation of this complex with direct molecular dynamics without doing any additional tricks. Now, if the dissociation would be the only slow process, that would not be such a big problem, because if we just know it takes 1 hour and we know everything else, like all the intermediates, the binding rates, etc., then you could argue, well, then in principle we have what we want, and we can just get this number of 1 hour from the experiment and we're done. Unfortunately, most systems are not so simple. Most systems have many intermediates. Many of these are long-lived. There may be many ways these 2 proteins can bind to one another that are at least metastable, so they have some stability, maybe not the most stable state, but they could still have lifetimes on the order of milliseconds or microseconds, and all of these create a lot of headaches when you are doing simulations because you get stuck there, and on your timescales that you can practically access, this is still forever. So when you have these multiple states, then you cannot equilibrate between them efficiently, and we have this sampling problem. Okay, so what can we do? So we can, of course, throw as much simulation power at it as we can to start with. With a modern graphics card like a GeForce 1080 or a Titan X, you get about 350 nanoseconds per day. That's actually for a smaller system, a 50,000 atom system. Okay, so for the 100,000 atom system would be about half of that. It is interesting to compare that number to the world's fastest supercomputer for molecular dynamics. That's the Anton 2 supercomputer at the Shore Research, which gives you about 70 microseconds per day. So, in other words, we need about 200 graphics cards to get the same throughput. And we can't get the same simulation lengths. This is giving us one trajectory that is very, very long, which is convenient if the total length is long enough so that we can really equilibrate over all the relevant states, because then it's relatively easy to analyze this data. Essentially, you can just compute regular averages. You can essentially count how often has the protein been in this or that state, how often was it bound or unbound, or folded or unfolded, or whatever. Here, this is not so easy, because we can generate the same throughput if we just put a lot of GPUs into a cluster. And it's even much cheaper to do that. Unfortunately, these trajectories here have not been started from the equilibrium distribution, because we don't know the equilibrium distribution in advance. So they all start from somewhere, presumably some crystal structure or some unfolded state, some starting state that we can easily generate. And that means we have to do something to the data in order to compute equilibrium quantities, which is easier here. This is easier here if the total trajectory length is long enough. The total trajectory length we can afford with the Anton 2 is maybe 10 milliseconds. If we run this for a couple of months, 10 milliseconds is still pretty short for most biomolecular processes. So I think even with an Anton 2, we kind of have the same problems, but there are certainly some systems we can sample to death with an Anton 2. All right, so we have this issue here, the sampling problem. We have these slow events, or these rare events. Independently of that, but especially if we have solved this problem, at least partially, we have an analysis problem. We have a lot of data, and we are trying to understand what is in the data, what are the relevant slow events, what are the relevant long-lived states. And this is something which is very difficult for biomolecules because they are simply large. There are a lot of things that can happen. Looking at huge amounts of data is not a very promising approach, and well, often people are analyzing big data sets by thinking of some reaction coordinates, some relevant degrees of freedom, plotting the trajectories on them, maybe doing PCA, things like that. And hopefully we will convince you in these couple of days that you can do much better than that. And finally, we would like to reconcile our simulation data with experiment. So the question arises, can we compute certain experimental quantities from the data? And how can we compare to experimental data? And if there is a disagreement, what do we do about it? And so there are also estimators that help us to actually do that, and that are based on the idea that our simulation force fields are now, after decades of development, good enough that they are not giving us pathetically wrong structures. In other words, the structures that you see are physically meaningful. Yeah, you don't get overlapping atoms, you don't get crazy dihedral torsion angles, you don't get clusters of ions where there shouldn't be any, etc. So the metastable states, the long-lived states, are probably right. It's probably right that there is something. but often the equilibrium between them is wrong, or is quantitatively not correct. It can be easily off by a factor of 10 from the right equilibrium or from the right rate, because there is an exponential dependency between the probability or the rate and the energy. Force fields are parameterized using energies. Factor of 10 on the probabilities or rates, that means less than 2 kT, less than 2 thermal units of energy in the energies, and that's not much. So it's a little perturbation in the energy means a large perturbation in the probabilities or rates. So it is to be expected that it's very difficult to be quantitatively correct there. So we need a way to somehow reweight our results and adapt them to experimental data, if we have an experiment that we can trust, which is again not trivial either. Okay, all right, so let's start with the sampling problem. The question that I'd like to address now is what do we do if we can generate a lot of data in short trajectories, but these short trajectories do not have the right equilibrium, or initially we don't know what the equilibrium should be. And the idea here is to use Markov models in order to fix this equilibrium. And this is actually the main reason why Markov models are used. So this is kind of the success story behind Markov models, because they just provide a very easy way of doing this. There are many other methods that do that too, but Markov models is essentially pretty much the easiest you can do to achieve that. Okay, so first of all, there should be a movie. It's not moving. Okay, this is not playing, I'm sorry. So what you should have seen is, this is just butane, a very small molecule, and it's basically flipping between 3 different torsion angle settings. So there is this torsion angle in the middle, and there's this bond around which there is a torsion angle, and the molecule can flip between 3 settings. These 3 settings are metastable or long-lived states. And so when you are within these rotamers, within these torsion settings, the molecule is fluctuating around on short time scales, and then there are rare events where you flip between the other to the other torsion angle settings. So this is a nice example of a metastable system. Metastability means almost stable. That means you have, in this case, 3 states that are almost stable, that are stable for a long time, and then there is a rare event which takes you out of the state. And based on this observation, we get the idea. Oh, now it works. Okay, right. So this is the movie to what I just said. So based on this, we may get the idea, well, maybe we can get a simple model of this phenomenon with 3 states corresponding to these 3 conformations and transition rates between these conformations. So, and the hope is that with this we can fix our problem with the unknown equilibrium. Okay, more formally, what is the problem with the unknown equilibrium? Well, so first of all, if we have molecular systems, molecular dynamics, then this is a stochastic dynamics. That's very important. Of course, there are Newton's equations of motion that you're integrating, but they are coupled with a heat bath, for example, or some algorithms which make sure that you are simulating at a certain ensemble, which is usually not the ensemble that conserves energy, the microcanonical ensemble, but some other ensemble, for example, an ensemble that conserves the temperature, or the temperature and the pressure. As a result of that, the data points that you sample from will, if you sample long enough, be distributing according to this probability distribution, this equilibrium distribution, which is the Boltzmann distribution. So it's e to the minus some energy. And this is normalized by the c, which is an integral, the partition function. And basically this is a high-dimensional integral, and u might be a complicated expression, so this is nothing we can actually compute. Now, many algorithms don't require us to compute C. For example, molecular dynamics and Markov chain Monte Carlo don't require us to compute C. They just allow us to sample from this quantity, but they do that by generating trajectories. And these trajectories take a long time to generate samples that are uncorrelated. Yeah, so in order to get two samples separated by some time, where you can say these are now two statistically independent samples of this distribution, you have to wait the longest equilibration time of the molecule. And the longest equilibration time of the molecule, that is the time scale, the slowest time scale of the molecule. So if you have a protein, often that is maybe folding or unfolding, essentially you have to wait until the protein folds or unfolds and unfolds once before you get a second sample that is actually an independent sample. So in other words, it takes you forever to sample from these distributions with the simulation methods. Okay, the quantities that we actually want to compute usually are expectation values. Expectation values such as this or this. This is the normal expectation value, such as, for example, the probability of being in a folded state for a protein. If we want to express this, we just take the equilibrium distribution, we multiply it with the value that we are observing, in this case just being in folded state or not. So we just define A equals 1 when we're in the folded state and 0 when we're not. And then this equilibrium average with this A will just give us the probability to be in the folded state. And likewise, we could compute the probability, or we could compute the expectation value of some fluorescence that is experimentally measurable, the expectation value of some distance, something like this. From this quantity, we can get free energies, like the free energy of folding or binding. And with this other expectation value here, we can compute dynamical averages. So this is things like rates, mean first passage times, escape times, all of this stuff. Correlation, time correlations, etc. can be expressed in this way. Okay, so it always ends up being the same problem. We have to compute these integrals. And this integral is something times the equilibrium distribution. And the problem is that in practice, we cannot really do this because our algorithms don't give us enough independent samples. So there's a very simple idea. We just take this integral here and we rewrite it in this form. So this is exactly equivalent. It's just rewritten a little bit. So we just say the integral is broken down into parts, into discrete parts. So we have a sum over discrete parts. Each of them has a weight, pi I. And this is now the integral over this region. So we just break down the state space into regions. And these are going to be our Markov states. And then this little integral, or this integral here, the integral over the part, we can just call that A dash. That is the expectation value over this region. So the expectation value is now just this discrete sum. And if we choose these regions well, so if we choose them to be like energy minima, or at least not cross to energy minima, then we can hope that within this region our simulation can sample quickly. So we got rid of part of our sampling problem here by breaking down, by dividing and conquering our problem and breaking it down into parts that can be sampled easily. But now, at this point, I just moved the problem to computing the pi's. Okay, so computing the pi's is still not so easy because this is a global equilibrium property. Right? So now I have the same problem here. I cannot hope to solve the problem just by mathematics, of course. I have to do something else. And this something else is introduced here. So this equation says there is another way to compute pi. And that is I can compute pi. Here I just write it as a vector. The t means it's now a row vector. I can write it in the self-consistent equation, which says pi is the equilibrium vector of some matrix P. So this is an eigenvector equation. Basically says pi is the eigenvector with the eigenvalue 1 of this equation, of this matrix. And this matrix, I can write it as this. But what it is simply the transition matrix of a Markov chain. So every element of this transition matrix tells us what is the probability to go from a certain state I to a certain state j in a time step tau. And the trick here is this is conditional probability. So this quantity here is again something which is easy to compute. All I need to do in order to compute this quantity is to be able to sample points x from this local region. So within this local region, I need to sample the points x with the right proportion, but it doesn't matter if I sample them in the different regions with the right proportions. And same here. So I have gotten rid of the problem of having to sample from the global equilibrium. That's the main point. Yeah? If I have like a two-well system with the wells A and B and they equilibrate slowly, folded and unfolded, bound and unbound, something with a high barrier in between, and I do not know in advance what is the probability to be in A and B, I can start some trajectories in A, some trajectories in B, and all I need is to count the number of times I go to the other side. It doesn't matter how often I actually started in A and in B, and how this number relates to the actual equilibrium. I can start 1,000 times here, 1,000 times there, whereas the actual equilibrium is 1,000 to 1, and I will still get the right estimate, provided that I get some transitions between them. That's the trick. Okay. He doesn't like this slide. Okay. Now, so that is the simple idea behind Markov state models. So I have this state space which contains metastable states where the equilibration between them is slow, and the idea is to break them down. However, so Markov state modeling has gone through several iterations, and the idea of how to best do it has changed a lot in the last years. So, in my opinion, the first real article on Markov state modeling is this from 1999 by Christoph Schütte, that actually ended up Christoph's habilitation thesis. And Christoph is now the president of the Zuse Institute, which is this building, more or less around us here, this building and this building. And so this basically is a mathematical article which lays the mathematical basis of the idea of Markov state modeling and using that to sample molecular dynamics. Then in the mid-2000s, people started to use and develop this idea further and to actually formulate methods and algorithms that could be applied to real molecular dynamics and to molecular dynamic simulations. Several groups started doing this in the mid-2000s, including my group, Vijay Panda's group, and several other groups. And basically, this more or less has been the time when computers started to be fast enough so that you could actually simulate at least small peptides long enough so that you could sample between the different conformations of these peptides. Our idea at that time was more or less what I described to you before. we have multiple metastable states, multiple conformations, and we divide them into substates, and these are the Markov states, and then we estimate a Markov chain between them. So for a couple of years we thought this is a good idea, until we figured out that there is a better idea. And this is based on the following mathematical insight. So this slide shows you something about the spectral properties of molecular dynamics. And this is a bit abstract, but I'm trying to walk you through. So molecular dynamics simulations are always about trajectories, right? So you start at a certain conformation, a configuration to be precise, so a certain position of atoms in the 3D box, and you run the MD algorithm for some time, and you get a trajectory in this n-dimensional space, or 6n-dimensional if you also take the velocities. But yeah, MD is always about these trajectories, and you don't really think very much about probabilities. At least the algorithm is not really formulated in a way that you think about probabilities. But this here is a description of the dynamics in probabilities. So here we do not think of one molecule evolving in time, but we think about an ensemble of molecules evolving in time. And this is the type of dynamics that you should think about when you do, for example, an experiment, like a temperature jump experiment would be of that nature. You have an ensemble of molecules, let's say proteins, in a test tube at a certain condition, and then you change the condition. For example, you heat up your sample with a laser pulse. And now, because these conditions have changed, the molecules like to change their equilibrium. They like to change the fraction of folded and unfolded. So they will gradually go there. Of course, every single molecule does whatever it likes to do all the time. It folds and unfolds. But if you look at the ensemble, the ratio of being enfolded and unfolded will change. This is what this equation describes. It says you start with some probability distribution of molecules, and you evolve it for some time, and you get another probability distribution of molecules. Why is this useful? Because a Markov state model is basically just a discrete version of this equation. This P here is just like the continuous operator behind the Markov state model transition matrix. So if you want to get a good model, meaning one that makes little error or approximates the real dynamics well, you somehow think about what do I need to do in order to approximate this operator well. Okay, so what is an operator and how do I approximate it? I guess at least some of you will know about quantum mechanics in the room, and for those it's helpful to think about this problem in the same way. You have an operator which has eigenvalues and eigenfunctions, and you get a good approximation if you approximate the most important eigenvalues and eigenfunctions. Okay, what does that mean? Let us look at this example here. This is a potential with 4 wells, and we imagine that we have just a diffusion in this 4-well potential. So we have a dynamics which runs around here in the minima, likes to stay in the minima, and occasionally we make a transition across one of these barriers. So the equilibrium distribution of this system will more or less look like this. We are most often in the minima, of course, and we are most often in the C state, and less often in B and D, and even less often in A. And very rarely we are on one of the transition states here, or barriers. Okay, but that's the equilibrium. Now, what is the dynamical behavior? So first of all, what we could do is we can look at these trajectories and we can ask if we are in a certain position, now, for example, position 25 here, and we wait a certain time tau, that's called lag time or delay time, And then, let's say, at a later time we were in position 50. Then we make an entry at this matrix here, 25, 50, that would be here. And we basically take a histogram over these jumps. And then this is what we get. So we will get, that depends now on the choice of the lag time tau, of course, but qualitatively for most lag times we will see this structure, that we have these 4 densities in this plot. And these densities simply tell us when we're in state A, so this is minimum, we are likely to stay in state A, at least as long as the lag time tau is not too long. As long as we're on a lag time where A is metastable, so if tau is shorter than the lifetime of A, then we will likely stay in A after that time. And the same for B, C, and D. But we already see at this matrix that it's easier to go from A to B and from C to D than it is to go from B to C. Because between B and C there's this disconnected region here. And that is because there's the high barrier here. And so all of this, what I've just described to you, is in these dominant eigenfunctions and eigenvalues. So this operator here has eigenvalues, and if I order them from the largest to the smallest, then the largest one is 1, and then I have 3 eigenvalues that are close to 1, and then I have a gap, and the rest is relatively small. And what that means is I have 4 metastable states. This eigenvalue, 1, just tells me there is a stationary process. So there is an equilibrium distribution that never changes. So that has nothing to do with the dynamics. These 3 guys that are close to 1 tell me that there are 3 slow processes connecting my 4 minima. And these eigenfunctions here, they tell me what is happening at those time scales. And let us first look at these. So these are 2 different representations of essentially the same sort of eigenfunction. They're just different. They just differ by a weighting. So this is constant. This is just corresponding to the stationary process. Not interesting. This eigenfunction here is negative at A and B and positive at C and D, or vice versa. You can just multiply it with some value. It's still an eigenfunction. So that means on the slowest time scale corresponding to this eigenvalue very close to 1, there is a transition between AB and CD. That means across this barrier. Okay, and that is obviously the slowest process in the system. And the next eigenfunction tells you there is a somewhat faster process, which is the transition from A to B and back. And then there is C and D, etc. So these eigenvalues and eigenfunctions decompose your dynamics into slow processes and faster processes. Now, of course, at some point it makes sense to make a cut, to stop resolving and say, okay, this is the slow term, this is the slow dynamics I'm interested in. Everything else is just fast fluctuations and details. And in some cases it's more natural than in others where to make this cut. I mean, here I have this obvious gap, so it makes sense to have a 4-state model. But you could, of course, do a 10-state model in this case. It will just give you something less intuitive, because then you start also resolving the insides of these barriers. Okay. And now, what does this have to do with Markov modeling? Well, because a Markov model is nothing else than a discrete representation of this operator here. What we need to do in order to get a good Markov model is to get a good approximation of these eigenfunctions. And that sounds a little bit metaphysical at this point, because you don't know the eigenfunction, so how does this help you? Well, it helps you to actually formulate algorithms that provably do that. And those algorithms will be what you're using. But intuitively, it's probably easy to see for a 2-well system that if you have the option of defining 2 states, then it's best to define them such that you divide the two minima at the transition state, at the barrier. That's just transition state theory. Right? But this Markov modeling theory tells you it's even better to use more dividing surfaces, to use more than two states, because you will resolve this eigenfunction better. And the error will be smaller. And indeed, the transition rate between the two wells computing from a Markov model that has more than two states will be more accurate than that from a 2x2 Markov model. And this is typically why, when we do Markov modeling, we do clustering into a bunch of states, let's say hundreds of states, even though in the end we don't really like to have hundreds of states. But this is just in order to discretize the problem. Okay. In the end, it would be nice to have a model with a few states, because that's easier to interpret. We would like to have, like in the butane case, we would like 3 states, which are the 3 torsions. Then we can look at the structures and immediately tell, oh yeah, those are the 3 states. And we want something like that for proteins too, where we see folded and unfolded states, or opened and closed, bound and unbound. So we need to do something else with the model in order to coarse grain it again. And so hidden Markov models is one way that allows you to do both. You start with a fine discretization, but then you find a coarse graining into a few long-lived states. And this is what you directly estimate with the HMM. So this will be one of the methods we will be using probably on Wednesday. And there is another method called VAMPnets that I think Luca and Andreas will be telling you about tomorrow night, right? Tomorrow evening. And VAMPnet directly gives you a Markov model. That means a normal transition matrix between different states, but the assignment of your MD frames to these states is fuzzy. In other words, some of these MD frames or some of these configurations will be clearly assigned to one of the metastable states, such as, for example, this dark red point here for in alanine dipeptide. This is a 5 psi angle for alanine dipeptide. But there are some states here at the boundaries which are fuzzily assigned, so they do not clearly belong to any one of the metastable states, but they are somehow a transition state. And with this flexibility, you can get very high quality Markov state models, even if you use only a few states. But it's not so easy to find these assignments. You have to somehow have a powerful algorithm that allows to identify where are the transition states and how should these functions be defined, which assign configurations to states. And in VAMPnets, we do that with neural networks. All right. Okay, so what's the time situation? Okay, 15 minutes. So these were kind of some basic ideas how to do Markov modeling. So Markov modeling has become kind of a name of a family of methods. There is like the classical MSM or Markov state model, which basically means you have a configuration space which is divided into substates with some sort of clustering, and you estimate the transition rates between them. And then there are all these variants like hidden Markov models, Markov state models with fuzzy states, Markov state models with cores, with God knows what. So, so if you want to be precise, there are other methods that one should be using, other names that should be using, but Markov state modeling is kind of referring to the whole family of these ideas. Okay, in practice, if we build a Markov model, so and assuming we do not use something like a neural network, but we go via clustering, then we usually have the following problem. We start with a very high-dimensional trajectory, some protein in solvent, for example. Okay, now the first step usually is to throw away the solvent. You don't necessarily have to do that, but at least it helps to reduce the dimension a lot, and very often the solvent equilibrates relatively fast. Not always, there can be structural waters that may be important, but in many cases this is at least not bad. I usually also discard the velocities. This is, I think on the time space we're interested in, this is never bad because the velocities decorrelate in picoseconds, so that is not an issue. But then we still have thousands of degrees of freedom. Yeah, even just the heavy atoms of a protein, that may be thousands of atoms, so 3 times that in degrees of freedom. And if we directly cluster in this space, usually we get very poor results, because clustering very high-dimensional spaces is difficult. Because in a high-dimensional space, essentially everything looks like a ball, like a high-dimensional ball. So everything is sort of equally far and close. So it is very important to first reduce the dimension in a way which doesn't destroy what we're interested in. What are we interested in? The slow processes, or the rare events. So we want to reduce the dimension in such a way that we preserve the rare events. And this is where we come back to this, essentially in this theory slide where I explained to you the eigenfunctions and the eigenvalues are important. Well, we don't know what the eigenvalues and the eigenfunctions are, but we can show that there is a principle, a variational principle, that allows us to build an algorithm which optimally approximates these eigenvalues and eigenfunctions, even if we don't know them, and which only uses things we can compute from the trajectories. And that's called the variational approach of conformation dynamics, and basically it's a dimension reduction technique that will give you a subspace of your original space, and in this subspace, the coordinates in this subspace are the coordinates where the slow motion happens, or the rare events happen. And to give you a simple example, you have these 2 point clouds here. They are generated by a 2-state process, where we either sample from this Gaussian, from a Gaussian distribution which looks like this, or from a Gaussian distribution which looks like this, and we rarely switch between them. So this is the rare event, the switching between these 2 states. When we're in one of these states, we sample actually super fast, so actually uncorrelated. But because of this weird shape of these 2 states, it's a little hard to separate them. So if you just do like a principal component analysis, you will get this axis here as your largest principal component, because this is simply where the maximum variance is. But now if you project all the points onto this axis, so essentially along this axis, then you will mix these 2 slowly mixing states. So you will no longer see the rare event dynamics, right? So of course this is a made-up example. PCA is not always that bad. But in a high-dimensional system, like a protein, you often have many things that are just fluctuating with large amplitudes, but they're really not interesting at all. Like, for example, a tail or loops or something like this. So you can have lots of things that have large amplitude motion, but they're completely uninteresting because maybe you have a very localized rare event in your binding pocket of the protein. And if that's a small amplitude motion, it will be completely washed out by these large amplitude fluctuations. So going for large amplitudes is not necessarily a good idea. At least not if you want to build a Markov state model, because Markov state modeling is about finding the slow processes. So you want a dimension reduction technique that leaves the slow processes in and removes the fast stuff, independent of the amplitude. And this variational approach, or TICA, which is basically an algorithm using this variational approach, does this. So this will be one of the first algorithms we will be using, and here's just a simple example of this variational approach in action. So we take this 10-dimensional time series. We try PCA, okay, that gives us sorting of the dimensions to a large variance and small variance, but it still looks not very interesting. And if we use this variational approach, or TICA, we get this. So you see 3 coordinates now, where you really see a distinct transition behavior, some slow processes, and then this looks pretty much like noise. And now you can additionally introduce a scaling, essentially just multiply these vectors by the eigenvalues that TICA computes, and this is what you get. And this is the kind of dimension reduction you want. You are left with the slow processes and you have removed all the fast stuff. And this can be used, of course, for molecules too. So here is an example that shows you the 2 slowest processes computed with this approach for this peptide here, which is from one of the Anton simulations. And this coordinate here is like one of the coordinates along which you can define the folding. So you can see that the helix and the hairpin are folding along this coordinate. Okay, so now we are in a lower dimensional space. Now we can apply clustering. Once we have applied clustering, we can estimate the transition probabilities between the clusters. You will hear more about that, but basically what we do here is we estimate the transition probability with maximum likelihood. So the most probable transition matrix given the number of times you have seen a transition between the different clusters. Of course, there is still some uncertainty on this number because you only have so much data to estimate this quantity. So there is an uncertainty, and that should be estimated too. And then you have a Markov state model. So then you can start analyzing it. And you will see several ways of doing that. One way, for example, to do that is to apply transition path theory that allows you to compute transition pathways from some set of states A to some set of states B, like the ensemble of folding pathways, for example. You can ask what are the metastable states, so which of these clusters do I need to group together to get the long-lived states? You can compute experimental observables, and many tools to do all of this stuff are in PyEMMA. Okay. Maybe you want to directly use some method that gives you a coarse-grained model, like hidden Markov model, for example. That could give you, like, as you see in this example, this is ligand-protein binding, different ways how the ligand binds to the protein, unbound state in green, then different intermediates in blue, and the bound state in purple. And yeah, there's this review book we wrote a couple of years ago, 2013. It's not really super up to date anymore, obviously. Many things have happened since then, but it's still a good introduction, I think. This you know about, obviously. This is our GitHub page. So if you're using PyEMMA after this or even during this workshop, if there are any issues you encounter, so if you think there is a bug or something isn't working as it should or some documentation is wrong, you can post an issue here. Everybody can do this. You just need a GitHub account. You can, if you're really interested in development, you can set a watch. That way you will see what is going on the developer side. So when new features are added or issues are discussed, etc. If you like the package, you can give us a star. If you want to develop yourself, you should fork the repository, so you make a personal copy. You can play with it, and then you can play back a pull request with your own code, and then that will be reviewed and merged if it works. Okay, I guess I should. No, I have 5 minutes, so I'll quickly walk you through one application that we did. Just to come back to this problem that I started with, protein-protein association. This serves as an example in 2 ways. So in one way, it's a demonstration of what can be done, and in another way, it's also a warning of that you still need a lot of sampling if you want to describe slow processes. So again, this is a 100 nanosecond trajectory, and this just shows you a little diffusion of the 2 proteins towards each other. And so what we did here, or specifically Nuria, what she did here was the following. We ran many of these simulations. And basically, we simulated each of them for a couple of 100 nanoseconds. And then, based on all the simulation data we had, we selected candidates, so conformations that we thought are promising. In this case, we just wanted to explore, so we did a very simple clustering using RMSDs, although that is not necessarily the best metric to do that in hindsight. And then we selected configurations in clusters that only had a few samples. So basically, the fewer samples in a cluster, the higher the probability of being selected. With the idea we want to explore, we want to favor sampling in states that are still undersampled. And we just kept doing that in many iterations. And so what we see here is if we ask, if we start from the dissociated state with the 2 proteins separated in solvent, and we ask what is the distance to the native complex, then we see that in aggregate simulation time, so that's just the total simulation time spent in all trajectories up to a certain point, we see many of them somehow get closer and eventually bind to the native state. This is 0.2 nanometers or about 2 angstrom in a root mean square deviation, so this is essentially the same state. And we need about 10 microsecond simulation time for that in maybe 100 trajectories of 100 nanoseconds or so. So that's fairly easy to bind the 2 proteins. But the surprising thing is, with this strategy, we can also unbind the proteins, or dissociated proteins. So if we start from the actual crystal structure, so from the tightly bound state, after about 100 or 200 microseconds, we can see dissociation events. And that is surprising because, as I said, the lifetime of these states is an hour in equilibrium. So either the simulation force field is completely wrong and it's way too unstable, or we did something right. Okay, so we collected a total of 2 milliseconds of data like that, so that's a lot, obviously. We got thousands of events where the 2 proteins kind of associated and dissociated, and several tens of events where they really went to the crystal-like bound state and back. Okay, then we did this dimension reduction. So using the variational approach, discretized, using k-means, and built a hidden Markov model. And we went through many iterations of this because it was not so easy to find a good set of features that would resolve the things we wanted and not resolve too much, etc. So this, in hindsight, always looks easier than it is, so it took a while to do that. But so here is what we find. So first of all, we can compute certain quantities like the binding free energy, the association rate, dissociation rate. Now the association rate is in the right regime. It's very close to the an experimental quantity, so that's as good as we can hope it to be. And the uncertainties are small. The binding free energy is different, but it's not wrong because the uncertainty is huge. The same, this is dissociation rate. These two, of course, go together. It's still essentially a two-state process in the sense that dissociation is really slow, and this uncertainty directly translates to this uncertainty. So it's right within the uncertainty, but the uncertainty is large. So, okay, we were mostly interested in binding and binding mechanisms, so this is not necessarily a deal breaker. But still, even with this adaptive sampling, which allowed us to get dissociations, we didn't get enough dissociation events so that we can get small errors here. So you need quite a few events to get acceptable errors. But there are other methods which do this better, such as multi-ensemble Markov models that you will hear about on Wednesday, I think. And there we can combine enhanced sampling methods which really drive the system over high barriers with unbiased MD to get small errors on rare events. But okay, so anyway, this is what we get for Barnes-Basta. So we can now do a couple of analyses. So one thing we did is to compute free energy perturbation experiments. So we took our ensemble that we got for this wild-type Barnes-Basta simulation. Essentially, each configuration that we have sampled has now an equilibrium probability through the Markov model. And now we can ask if we changed these equilibrium probabilities, for example, by mutating the protein, how would the binding free energy change, or how would the binding rate change? For this, we need an additional simulation that simulates what happens if we actually do this mutation. And this we did with the implicit solvent force field. So we just took all of these configurations, we exchanged an amino acid by an alanine in this case, recomputed the energy, that gives us a new weight change for this configuration, and then we again compute the binding free energy or the binding rate. And this is something we can compare with experiment. And this gives us a lot of points to compare with experiments. Okay, so the overall picture that we get is the binding happens more or less like this. We start from the dissociated state. Then there is an ensemble of states where Barnes and Basta are somehow sticking together, but not in the right orientation. Then Basta can enter the binding groove of Barnes, but in this state, for example, is upside down. In these states here, it's kind of in the right orientation, but not exactly. And then eventually, they find the right configuration. In this state, they form a certain contact, which is an important transition state contact. And then they simply go together, essentially, very rapidly. And then there are still two states here, between which the complex can fluctuate between. And okay, first of all, we get this coarse-grained Markov model, which basically says there are these groups of states and they have certain transition rates. And so this is something that you can show the experimentalists, and they will be happy to see that because they can normally parameterize like 3 states with their experiments and only see 5 states, so there is something additional. But now, because you have these states and you can really look into the structural details, you can ask, so what is specific to these states? So for example, in this pre-bound state, this transition state, you see that there are 2 particular amino acids that form in contact here, and this is like an anchor which connects two proteins. And this is essentially the only connection between them. Otherwise, the state is really floppy. And this is interesting because it's known that this residue here, this arginine, if you mutate it, then the association rate decreases dramatically. So this explains structurally why this happens. And if you look at these two substates in the native state, then you see, well, they are looking not too different at the first side, but first of all, this state, the larger state here, or the more popular state, that is essentially exactly like the crystal structure. This state is a little bit different, and you see that this helix here is rotated to the right. So basically there is a little rotation of the two proteins with respect to one another. And then looking at these side chains, you see that, for example, this tryptophan here gets exposed, and then this again is something you can tell to experimentalists and say, okay, if you can do tryptophan fluorescence depends on the solvation states of the tryptophan. This is a microsecond process, so if you do like temperature jump spectroscopy or fluorescence correlation spectroscopy with tryptophan fluorescence, you should see a process in the microseconds. So, and this finally is just a movie which is generated from the Markov model. So basically we take the Markov model, we generate a process which jumps between the different states, and then we go back to the trajectories and we just patch together trajectory pieces. So this shows you like a 100 microsecond trajectory generated this way. Now I cannot get back to it, which gives you an impression of how this process happens. I don't know why I can't play it twice. Anyway, so that was it, and yeah, I guess I've already introduced at least the people that are involved in the PyEMMA workshop. So just one more thing, we have some room for new PhD students and maybe also postdocs. So if you're interested, just drop me an email. Otherwise, and of course, master project as well. And otherwise, thank you for your attention and have fun during the workshop.